All right, guys, welcome back to Biology with Mrs. R. We are going to talk today about natural selection and speciation. So let's dive in. So we are looking at Georgia Standard of Excellence SB6, and we're focusing on B and D today. So SB6, of course, is assessing the theory of evolution, and B has us explaining patterns in biodiversity that result from speciation. And has us looking at explanations of how undirected genetic changes in natural selection and genetic drift have led to changes in the populations of organisms. So we should be able to explain how natural selection takes place. You should be able to explain the different models of natural selection, including directional selection, stabilizing selection, and disruptive selection. And you should be able to demonstrate an understanding of speciation and how environment can affect the formation of a new species. So the first thing we need to do is talk about natural selection. And before we actually talk about the three types, we need to know that natural selection, like they said in a video that we watched previously, natural selection is just where organisms with traits that are better suited to the environment survive and reproduce. And when they reproduce, they pass their genes on. So as far as types of natural selection, we have stabilizing selection, directional selection, and disruptive selection. So stabilizing selection is where individuals in the middle, the ones that have the middle trait, are favored over either extreme trait. Directional selection is where either end of the extreme are favored over the individuals in the middle or the other extreme. And disruptive is where both extremes are favored over the middle. So I'm going to give you some more information on that, but let's watch a video first.
Natural selection is the gradual process by which heritable traits become either more or less common in a population as a result of any or all of the following items: overpopulation, competition for resources, changes in the environment, and predators. There are three types of natural selection: directional, stabilizing, and disruptive. Directional selection occurs when one or two of a trait extremes are selected. A classic example is the peppered moth during the Industrial Revolution. In the mid-1800s, most pepper moths were white, and black pepper moths were rare to see. As the Industrial Revolution moved forward, factories were fueled by coal, and many of the trees became dark. By 1900, the population of pepper moths were mostly black. The black color on the dark tree was more favorable than the white color for the peppered moth. Stabilizing selection occurs when the environmental factor selects for the average of a trait. One example is the number of eggs a bird population lays. Many birds may lay 2 eggs, but this may not be enough for the birds to survive, and 4 eggs may be too many birds to feed. Over time, the population will stabilize around 3 eggs. Disruptive selection is the opposite of stabilizing. The average is not favorable and the extremes become favorable. For example, you have a population of rabbits that live along black and white rocks. The fur color of the rabbits range from white to gray to black. The white and black are the extreme colors, because the white and black colors can camouflage on the white and black rocks. The extremes will be selected for, and the average color will be selected against. Over time, this type of selection can lead to speciation. Thanks for watching, and remember, kindness multiplies kindness. Be kind to someone today.
All right, guys. So there's some pictures that kind of just go over what he was just saying. So directional selection is where one of the extremes are favored. So here we've got some lizards. And in these lizards, long wiggly tails look like a snake and they scare predators off. The longer the tail is, the more it looks like a snake. So the more likely it is to survive and reproduce. That's directional selection. Stabilizing selection is when we kind of are selecting for the average or the moderate trait. So for instance, with this one, short tails mess up cats' balance, but long tails drag on the ground. So the best option for cats is medium tails. And then there's disruptive, where it's choosing Both extremes are selected for. I don't want to say choosing, but both extremes are selected for or are better fit in the environment. So, short tails help keep predators from catching you. Long tails are good for balance in the trees. Medium tails don't help with anything. So, the medium tails in these squirrels are not going to be the ones that survive. The ones that survive are going to be the short tails because they don't get caught by predators, or the long tails because they're able to balance in trees better. So, next up, we're going to talk about speciation.
Why are the animals in Australia so unique? You've got the kangaroo and wallaby, marsupials that carry their young in a handy pouch, the cute but lazy koala, and the weirdest of all, a beaked egg-laying mammal called the duck-billed platypus. In this video, you will learn how these weird and wonderful animals are formed in the process of speciation. Speciation is the formation of a new and distinct species in the course of evolution. Many millions of years ago, the continents that we know today were all joined together in one supercontinent known as Pangaea. Over several hundred million years, these continents slowly drifted apart from each other. When Australia finally separated from the other continents, its animals and plants were cut off from those on other land masses. This isolation is the first step in the formation of new species. It splits a species into two different gene pools. As the animals and plant groups on the Australian continent drifted away from their relations, they became isolated from other populations of their species, because the sea acted as a barrier. Each gene pool was now exposed to different conditions, like climate, food, and competition. This is the second step in speciation. Some animals and plants that were not well adapted to the new conditions died, whereas those that were best adapted survived. Through the process of natural selection, the gene pool changed and the organisms became more and more different from those left behind. This is the third step in speciation. Eventually, these differences became so great that isolated populations are unable to interbreed and a new species is formed. This is the final step in speciation. There are many other examples. The lemurs and baobab trees in Madagascar that became isolated from continental Africa, or the famous giant turtles and unique flightless cormorants of the Galapagos Islands that were isolated from South America. Their ancestors arrived either by flight, swimming, or on natural rafts from the nearest mainland. Some of the species presented here are highly specialized. If their environment changes due to human or natural influence, they may become extinct, like the Tasmanian tiger, a stripy dog-like animal which became extinct when humans entered its habitat and started overhunting it. These are examples of speciation happening because of physical barriers. Members of the same species can also be separated due to biological or behavioral barriers. Birds of the same species, but living in slightly different areas, may over time develop different mating rituals or different mating songs. And so, the females are only attracted to rituals of the males from their area. Similarly, species living in different areas may face different climates or different food pressures, and so their mating seasons may end up being out of sync with other members of their species. This happened to two species of toad in North America. Over time, both of these resulted in speciation and are known as reproductive isolation. So remember, the amazing variety of flora and fauna that we have in our world is due to the process of speciation, the formation of a new and distinct species in the course of evolution.
All right, you guys. So, speciation is just when we get a new species. So, that's one of the big things that Darwin studied on the Galapagos Islands were the different species of finches. So there's a few different ways that speciation can occur. There's temporal isolation, and that's just where organisms mate at different times. For instance, there are two different species of trout. One of them spawns their eggs and sperm in the spring, one of them spawns their eggs and sperm in the fall. If the egg and sperm spawn at totally different times, they have no chance of mating with one another. Um, Another example is geographic isolation. This is where the organisms are physically separated. So this is what happened with the finches on the Galapagos Islands. They were physically separated from one another, so they didn't interbreed with one another, and they all began to eat different foods on their different islands. And then the last one is behavioral isolation, and that's just where the organisms have different mating behaviors. So it may be like the songs here that you can see, the bird songs. It might be that one of them is singing country and the other one likes rap, or whatever the mating song happens to be. In addition to speciation, there's different types of evolution as well. There's convergent evolution, divergent evolution, and there's coevolution. So, convergent evolution is where organisms are not closely related to one another, but because they live in the same ecosystem, they evolve similar traits. So, an example here would be wings in bats and moths. So, convergent, not related to one another at all, but because they live in the same area, they have developed similar traits. Divergent, on the other hand, this is where a species is one species, and because of different factors, they have diverged or split into several different species in a relatively short period of time. So, the big example with divergent evolution is going to be Darwin's finches. And then we have coevolution, and that's just where different species evolve because one species evolved and changed, so another species evolves with it. So they're like co-captains of a team. They are both working together. And so an example of this would be like bees and flowers. When flowers change, the bees change with them, and vice versa.
Thanks for stopping by. Today we're talking about convergent versus divergent evolution. This video was suggested by one of my subscribers, so here you go, Nekla, and I'm sorry if I pronounced your name wrong. And if any of you have any video suggestions, please throw them in the comments. Let's talk about divergent evolution first. So in divergent evolution, two or more distinct species share a common ancestor from which they both diverge. A common example is the modern elephant and the woolly mammoth. They share a common ancestor and yet evolved into two different species. Another example is the dog, the wolf, and the fox. Again, they share a common ancestor yet evolved independently, right? They all diverge from a common ancestor. Now let's contrast this with convergent evolution. In convergent evolution, two or more distinct species share traits not due to a common ancestor. So let's take birds, bats, and butterflies, for example. All have wings that allow them to fly, but they don't share a common ancestor with wings. So each one evolved to have wings independently. Right? Evolution converged on a similar trait in vastly different species. Right? So their wings are not due to a common ancestor. All right, so let's recap. Divergent evolution is when descendants of a common ancestor evolved independently and uniquely, like the dog, the wolf, and the fox. While convergent evolution is when species share a trait not due to a common ancestor, such as birds, bats, and butterflies' ability to fly. And that's it. Check out my video on analogous.
All right, you guys. So next up, we're going to go over a couple of vocab terms. So one of them is genetic drift. Genetic drift is just the change in allele frequency due to random chance events. These are things that don't have anything to do with natural selection. And genetic drift tends to work best in small populations. There's two things to look at with genetic drift. That is undirected change and directed change. So undirected change is when we have evolutionary change that is random. It's not guided by traits, by an advantage or disadvantage of a trait. Directed change, on the other hand, it's non-random evolutionary traits. Directed change is where we have our example of natural selection that we're thinking of, because it is directly caused by the advantage or disadvantage that an organism has, that a trait has to allow an organism to survive. Let's check out a video real quick on bottleneck and founder effect.
I know that a lot of students think that founder effect and bottleneck are the same because they both result in genetic drift, which is the random change in allele frequencies from generation to generation.
Although both modes of genetic drift ultimately have the same result, the two processes of how they came to be are completely different.
Bottleneck occurs when a population is reduced down to a few individuals, for example, due to a natural disaster like a flood.
Something that helps me to remember is to picture a bottle and visualize how the neck gets smaller and smaller as you move from the widest part of the bottle to the top.
Meanwhile, founder effect occurs when a small group leaves the main population to go and establish a new population. And remember, although the focus is now on the new population, the mainland population still exists with all of the initial genetic information.
As we just saw, both processes lead to genetic drift, but how we got there given each process was very different.
I hope that this video helped to clarify the topic of bottleneck versus founder effect.
Now, here's a short quiz.
All right. So with this, these are both ways that genetic drift happen, but bottleneck effect is when there's some sort of an environmental event, like a natural disaster, that leads to a severe genetic drift because it kills off a large part of the population. Founder effect, on the other hand, this is just where a small group branches off from the original population to establish a new one. And so one of the things that makes me think of founder effect, if you're from any sort of a small town, there's probably lots of different churches in your small town. Oftentimes, that's because there was one church or maybe two or three churches, and over time something happened and a group branched off and created a whole new church. And that's an example of founder effect type of an idea where it's where a group of individuals branch off and form their own little colony type thing. And then we have two last terms, and that is sample size and allele frequency. So, sample size is just the number of individuals or alleles measured in a population. This is really critical when we talk about the effects of random chance. Then we have allele frequency, and allele frequency is just how many times do you see that allele? So, if we look here at this first population, we see four A's and we see four B's in our population. Over here, we only see two A's and we see six B's. So that is just how many times does that particular allele show up in the gene pool? Is the population small? Does it show up a lot? Does it show up a little bit? How many times does that gene show up? And that is everything that I have for you for today's flipped video. We will talk about this more in class, and then we'll jump in and start talking a little bit more about natural selection and biological resistance and things like that.